So the second concept is about transcription, translation and translocation, right? So before we start with this, you should know the basic structure of DNA and various types of RNA. So DNA consists of two strands and the two strands are interwoven like this. So DNA has a helical structure. If you open up these two strands, they will look like this, right? So these two strands, they are in anti-parallel direction. If you mark up this strand as 5 to 3 and another strand will be in the opposite direction, 3 to 5. So the two strands are anti-parallel to each other, and DNA has four nucleotides, A, G, T, C, right? And you can write over here A, T, G, C. A always bind with T, T binds with A, G binds with C, and C binds with G. So this is governed by Chargaff's rule. Chargaff's rule, right? The two strands consist of complementary bases. A is complementary to T, G is complementary to C. A and D, they are bound by two hydrogen bonds. G and C, they are bound by triple hydrogen bond. So this overall structure, which has two strands, they are stabilized by hydrogen bonds between these complementary bases, right? On the other hand, various types of RNA. First of all, what is the basic structure of RNA? RNA always has a single strand. It is a single strand molecule, and it is made up of 4 bases. But instead of thymine, RNA has uracil, U. The rest 3 bases, they are same, right? So various types of RNA are, one is mRNA, second is transfer RNA, and third type is ribosomal RNA, right? M stands for messenger RNA. T stands for transfer RNA. R stands for ribosomal RNA. Now next is explain the meaning of genetic disorder and give names of some disorders. So what do you mean by genetic disorder? The disorders which are caused due to the change in the structure of the gene or due to the change in chromosome number. So what are the few names for these disorders? You can write down. It is Down syndrome, which is caused due to the trisomy of chromosome number 21. It is Klinefelter. It is sickle cell anemia. Sickle cell anemia, right? So next concept in this is central dogma. What do you mean by central dogma? Now, as I previously said, gene, they code for proteins, or you can say gene, they make protein within a cell. But how do they make protein? A gene will first of all produce a copy of RNA, and this RNA will get translated into protein. So this is the central dogma, which means within every cell this process is followed. First of all, this gene is changed into RNA. This RNA is then translated into protein. There is no way that gene can directly produce a protein or RNA can directly produce a protein, right? So this is a strict principle which is being followed within a cell. So the formation of RNA from gene is known as transcription. So this is the first process. And what kind of RNA is produced here? This RNA is messenger RNA, right? And then the formation of proteins from messenger RNA, this process is known as translation, right? Now we will see in the detail with the help of this diagram. We'll try to understand from here. Now, the first process, which is transcription, that take place in the nucleus. If this is the nucleus, this process take place within the nucleus of the cell. So as you know, DNA is made up of 2 strands, and RNA, as I have said, RNA is a single strand structure, right? So how RNA is produced from DNA? RNA is formed, this is RNA, it is formed complementary to one of the strand of the DNA. So these are 2 strands. If you focus on this strand, a molecule of RNA is being produced which has bases which are complementary to these bases on the DNA. If this is T, RNA will have A. If this is A, RNA will have T. But you already know, instead of T, RNA has U. If this is C, RNA will have G. If this is G, RNA will have C. So these are complementary bases or the complementary strand which is produced against one strand of the DNA. Now, once this RNA is formed, this RNA is the messenger RNA. Why it is known as messenger RNA? Because it carries the information of the gene. So this will exit out of the nucleus. In this picture, you can see the strand of RNA, mRNA is coming out of the nucleus through the nuclear pores. These are nuclear pores. So translation, it takes place in the cytoplasm. Once mRNA is formed, as you can see in this picture, the mRNA, once it is transcribed from the DNA, it will exit out through the nucleus. So these are nuclear pores. These are nuclear pores, right? Now once mRNA is formed, so you know now your mRNA has certain set of nucleotides, right? So these are the nucleotides. So 3 nucleotides together, triplets of bases, they are known as codon. So this is codon 1. This is the next 3 nucleotides are another set of codon. This is codon 2, right? Next 3 bases are codon 3 and so on. So the triplet of bases on mRNA, they are known as codon, right? Now, in cytoplasm, you have a cell organelle which is ribosomes. So these ribosomes, they help in protein synthesis, or you can say they help in the process of translation. How? First of all, these ribosomes, as you can see, they will bind to mRNA. And these ribosomes, they provide the room, they provide a place, a room where transfer RNA, they can come and bind with the mRNA. If there is no ribosomes, tRNA, they can't bind to the mRNA, right? So ribosomes, they provide a site for binding of tRNA. Now what are tRNA? T stands for transfer. So what do they transfer? They transfer specific amino acid. They transfer specific amino acid from cytoplasm to the mRNA. And how they do it? You can see the structure of tRNA over here. The one end is bound with this specific amino acid. So this amino acid, the kind of amino acid charged with tRNA, will be decided by the sequence of the codons. Say for example, codon 1 here is specific for the lysine. So tRNA will pick lysine. Lysine will bind to one end of the tRNA, and the other end of the tRNA will have a complementary basis to codon 1, right? So this is how this tRNA with lysine, it will bind at this position. So these complementary bases on tRNA, they are known as anticodons. So these are codons. Codons are the triplet of bases on mRNA. Anticodons are triplet of bases which are present on tRNA. Anticodones, they are complementary to codones, right? How tRNA bind to mRNA? With the help of complementary bases. There is hydrogen bond between these complementary bases, right? So by this way, another tRNA will come and occupy this site. Now you can see there are two amino acids adjacent to each other. A peptide bond will form in between these two amino acids, and this uncharged DNA, it will exit from the system. It will exit out. Right? Now this ribosome will shift in the forward direction. So this shifting of ribosomes, or the movement of ribosomes over the mRNA, is known as translocation. So in this way, there will be another tRNA and a sequence of amino acids will be bound together with the help of peptide bond. So what is this? A long chain of amino acid is basically known as protein. So this is how proteins are produced, and this whole process is known as translation, right? So briefly, I'll re-explain you the all concepts again. So what happened? Let's see. So this was the DNA. Once this is a strand of DNA. It has a sequence here, A, T, G, C, A. A mRNA will be produced which will be complementary to this strand, right? The strand which is in direction 3 to 5. Now RNA will be, instead of T there will be uracil, here it will be A, complementary to G will be cytosine, complementary to C will be G, and complementary to A will be uracil again. So this is a strand of mRNA which is produced from DNA. So this process is known as transcription. This takes place in the nucleus. Now once mRNA is produced, mRNA will exit out of the nucleus into the cytoplasm. Within the cytoplasm, there are ribosomes and tRNA, right? So you can you can write more bases over here. Just you can put some more bases for the understanding. Now, so these triplet of bases, they are known as codones, and they are specific to some amino acids, right? Say for example, UAC, they are specific for methionine. GUU, they are specific for amino acid valine. GAC is specific for an amino acid, say for example glycine. So all these amino acid, they will be joined together. A polypeptide or a long chain of amino acid will be produced. So this long chain of amino acid is known as protein. So this process from mRNA protein is produced, this process is known as translation. And translation takes place with the help of ribosomes and transfer RNA, right? And this step takes place in the cytoplasm, right? And this whole transcription and translation together is the central dogma.